Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, welcome you all to the third lecture of Response to Infection and Pathobiology course. And today's lecture is about bacterial structure and growth. This is Dr. Zaim Al-Jabri with you. Now let us move to the next slide. So in this slide here, what you can see, this is a picture actually of a handprint on a petri dish. So what we mean by a petri dish, it's basically like a plate with some food on it. So it's the same concept. The bacteria needs food to grow. So this mother, what she did was, apparently she's a microbiologist, otherwise she wouldn't have thought about it. So she brought her child's handprint and she printed it on top of a plate, and then she left it to grow for at least 24 hours or even more for this one. And what you can see here is small dots on top of that plate, and all of these have different colors and shapes and elevations and morphology, we call it, as you have been introduced to these new terms in the previous lecture. And what I wanted you to see and appreciate from this slide is basically that this child has these microbes, mainly bacterial microbes, on his hand, which is totally normal and accepted from a child who is playing outside. But not only children, actually all of us have some amount of microbes on our hands and skin and also some organs inside. And we will go through these into details in the later coming weeks. So now let us move to the next slide. So the objectives of this lecture is to describe some main structures of bacterial cell, and we go through a procedure called Gram stain, which is a very important procedure we do in microbiology for differentiation of bacteria. And then we move on to describe the process of bacterial growth and requirements for growth. So as this picture looks very, very complicated, but I don't want you to get very much worried about the details, but I want you to focus here. On the left-hand side is the bacteria, and then the archaea, and then the eukarya. And these are the three main domains of life. And what we cover in this course is just a small amount of bacteria, not all. And you can see that the branching of these bacteria, although it has gram-positive, gram-negative, some other bacteria like cyanobacteria, we're not going to go through these, but we are going to only discuss these medically important bacteria. And bacteria is different from the rest of the microbes and also from other eukaryotic creatures, and we are going to address this in the next coming slides. So in this slide, I want you to understand the basic concept of prokaryotic and eukaryotic cell, and also to appreciate the difference. And also you can see this very clearly from these pictures. The prokaryotic cell is totally different from eukaryotic cell in terms of structures and complexity. And I'm sure that you have gone through the basics of these cellular structures when you were at school, and also during maybe biology, you had previous biology courses. And I have made this table for you so that you can appreciate these differences. So if we first address the nucleus, or look at the nucleus in the prokaryotic cell, and we compare it to the eukaryotic cell, actually there is a major difference, because a nucleus of a bacteria, for example, will only have some DNA, but it will not have a nuclear membrane, or a membrane that goes around that DNA, while a eukaryotic cell actually have a very complex structure called nucleus. In bacteria we call it nucleoid. And the second point is the DNA. Actually, in bacteria, it's just a single chromosome, and what you see is a supercoiling or wrapping up of all the whole genetic material. While in the eukaryotic cells, we have multiple chromosomes, including human beings, that we know that we have a set of chromosomes of about 46 chromosomes. Now we come to the third point, which is the size of a ribosome. So what is a ribosome? Maybe you already know that the ribosome is simply a factory in which proteins are made or synthesized. So in bacteria, as a living being, of course, it will need to synthesize protein structures. And this is done in a structure called a ribosome of 70S size, while a eukaryotic cell have an 80S ribosomal size. The fourth point is respiration. By respiration, we mean breathing. So even the bacteria needs to breathe, and this happens via the cytoplasmic membrane. So the bacteria actually synthesizes this energy through the membrane. And if you block that membrane by, for example, giving the antibiotics, you will definitely kill that bacteria. The same thing for eukaryotes, like us as human beings. We don't live without energy, and the factory for synthesis of energy is called mitochondria, and these are those small organelles or orange organelles you can see from the picture. We have many mitochondria in each cell. The last point is the membrane-bound organelles, and what we mean by membrane-bound organelles are the small organelles found inside the cell, and they are closely related to the membrane. There are none in a prokaryotic cell. While in eukaryotes, they have plenty of organelles like Golgi apparatus and endoplasmic reticulum and so on. Now let us move to the next slide. So what you see here also, not only bacteria different from eukaryotic cells, but also bacteria are different from other microorganisms. As you know, that microorganisms can also be fungi, which means small, also mold, like mold and penicillium. These are fungi. Protozoa like parasites and amoeba, algae, and viruses. So all of these are microbes, as you can see in this picture here. But let us go through the main differences also through this table. So bacteria is the second column here. It's a cellular organism, just like fungi and protozoa, and they are all different from viruses because viruses basically are genetic material that do not have a proper cell. In this course, I'm going to teach you, along with Dr. Mihir, the bacteriology and mycology or fungi section, while viruses and protozoa are going to be covered by other tutors. Now we come to the nucleic acid. So bacteria have both DNA and RNA, similar to fungi and protozoa, while viruses should be either RNA or DNA viruses. Now we'll move on to the nucleus. So viruses have no nucleus because the genetic material is only dependent on entering into the host cell and then replicating within the host cell, while bacteria is actually a prokaryotic nucleus, very simple structure. Fungi and protozoa have a eukaryotic nucleus that we discussed in the previous slide. Now we move on to the outer surface. So bacteria have very rigid cell wall that we will discuss in the few slides next to this one, and it contains a peptidoglycan layer, while in fungi it's also a rigid cell wall, but it's mainly a chitin layer, and protozoa have a very flexible membrane. Viruses, on the other hand, have protein capsid and lipoprotein envelopes. The second point is replication. So bacteria replicate by a mean of binary fission or division, while the others, like fungi, they reproduce by budding or mitosis. Protozoa, they reproduce by mitosis as well. Viruses replicate actually by multiplication. Motility means the ability of the organism to move. So bacteria, not all bacteria can move, and we will learn that in a minute. While most protozoa can move, fungi and viruses are non-motile, or they are not able to move. So let's now move to the next slide. I'm sure that you have gone through the classification of bacteria in the previous lecture, and I don't want to go through this again, but I want you to appreciate that we can actually classify bacteria using different methods. The first one is based on the shape. So bacteria will have different shapes like you discussed in the previous lectures, spirilla, bacilla, and also cocci. And you can also have another classification on gram stain. And actually, your gram stain also can provide you the shape of the bacteria, but we call it either gram positive or gram negative based on their color. And the third classification is through oxygen demand. So usually we call a bacteria aerobic if it's requiring oxygen for growth, while anaerobic means the bacteria will be able to grow in the absence of oxygen. In fact, oxygen can be toxic for this bacteria if it's present in this environment. Now let's move to the next slide. Okay, so why do we need to perform gram stain? So as you learned in the previous lecture, gram stain is a very, very important, simple, and also easy method that we use in our microbiology lab. In fact, almost all of our specimens we receive in the lab, we have to go through gram stain. Why? Because gram stain, very simple and inexpensive, and it can tell us a lot of information. The main reason we do gram stain is identification of the bacteria, and the second one, it also helps us, that identification will give the bacteria its name, and then we can choose the proper antibiotics by doing the antimicrobial susceptibility that you have also been introduced to in the previous lecture. Again, we will see a lot of more examples of gram stains in the practical of this week, in the introductory week, and also you are going to see a lot of gram stain pictures in the next few weeks. So you need to remember something, which is bacteria cannot be seen by gram stain. There are many, many bacteria that we can see by gram stain, but also there are a number of them which we can't. And actually this is what makes our life harder, because once bacteria cannot be identified by a gram stain, it will tell us that this is a difficult bacteria. And I'm listing here a few examples, and you will learn each one of these bacteria actually later in this course, and I'm not expecting you to learn these details at this stage, but I want you to remember that these bacteria that we cannot stain with a gram stain, there is a reason why we cannot stain it, and there is also a possibility that we can use an alternative stain, which is a special type of stain to stain all of these bacteria. So you can go through this table, read it, but then we can discuss it later on, because we are going to study each of these bacteria later on in details. Now we move on to the next slide. So in this slide, I'm going to go through briefly the procedure of a gram stain. And usually we show it to the students when they used to come to the lab, but never mind, I'm going to also post some videos maybe later on when the time comes for you to see how this procedure is done. The first step is actually when we apply a stain. So what a stain means is it's basically a concentration of a dye that will give the bacteria a specific color. So we bring a glass slide and we apply some bacteria or a drop of the bacteria on top of that slide. And then we add the dye, which is the crystal violet here on the left hand side. You can see the crystal violet stain. We leave it on that glass slide for about 1 minute. We wash it with water, and then we apply iodine. As you all know, maybe that iodine is an orange color, not really brown, but it's orange in color. And the idea of adding iodine is that it binds to crystal and then it forms a complex. At this stage, all bacteria will look blue in color. Then we leave it for another 1 minute and we wash it. After washing it, we add alcohol. And the purpose of adding alcohol is to wash any excessive dye. So gram negatives will wash out this alcohol will wash out all the dye, while gram positive, because they have a very thick cell wall, as we will learn in a minute, it will actually retain that crystal dye inside. It will not allow it to escape. And then because gram positives are purple in color, but gram negative at this stage will not have any color, we need to add additional color. At this stage, of course, also we do not know which bacteria we have because we have not yet put it under the microscope. So we add another dye, like in the last step, we add safranine. Safranine is another staining dye that will add color, that now will color the gram negative bacteria. Now let us move to the second slide. So this is after staining the bacteria, you put it under the microscope, and this is what you are going to see. And when you learn this, I'm expecting you to be able to tell me that this is a gram positive because it's purple in color, and you can tell me also the shape. So the shape of the bacteria is cocci because they are round, and they are arranged in clusters. So we can call it now gram positive cocci forming clusters or in clusters. This is another picture, although the background is a little bit yellowish, but these are also gram positive cocci, but this time the arrangement is not clusters, they are actually in pairs or in small or short chains. This is a third picture. You can see the small dots inside the cells here in this slide here. You can see it's very small, tiny little dots inside the cell. So these are intracellular bacteria, and we call it gram negative cocci because they are staining pink or red, not purple. The fourth picture here is gram positive again because it's purple, but it's cocci in chains, not in clusters like the first one, not in chains or pairs like this one. It's actually longer chains. So these are a few examples that you will be able to see in the lab in the next few weeks. Now we move on to the next slide. So in this slide here, you can see a different story. You can see a bacteria here that are a little bit pink in color, but the background is blue. But this is actually not a gram stain. So if you can see, there are a little bit of a difference because this actually one of the bacteria that we cannot stain with a gram stain. We call it Mycobacterium tuberculosis, and we call this stain a Ziehl-Neelsen stain. And this bacteria causes the respiratory or the disease tuberculosis. Now we move on to the next slide. So you can see here from this picture, what I want you to appreciate is this is the bacteria here. So this is the relevant size of a bacteria. And what you see here is the virus. And here are different structures like proteins and atoms. While here we have the eukaryotic cells. So bacteria in relevant to other organisms is 1 micrometer in size. It's very tiny, and that's why you need a light microscope to see it. Anything below that actually needs an electron microscope with a higher magnification power to be able to see it. Anything below that or above that, sorry, it will be appreciated by light microscope. Okay, so let's discuss this slide here. It's a basic structure of a bacteria cell. So what I want you to really understand from this is that we have a lot of structures inside the bacteria. Some of them are called essential parts, which means that they all present in all bacteria, while others are non-essential parts. So please go through this list and read them. I'm going to go through almost all of them in a minute. The most important structure that I need you to appreciate first is the cell wall, and that what makes bacteria either gram positive or gram negative. The complexity of a cell wall is what gives the bacteria its structure. So, for example, can you see how thick is the gram positive wall compared to the gram negative wall? This thickness is actually contributed by the cell wall, and all of this complicated cell wall is actually called the peptidoglycan layer or a murine layer. So murine or peptidoglycan is the same word for the same structure, and it's very thick in gram positive cells, while in gram negative cells it's very thin. Here it is, this thin layer in between. And in gram positives, it has some specific structure called teichoic acid. You can see these hair-like structures on top, and also these ones, lipoteichoic acids. They are not present here. What you see here is a different structure. It's all antigens. And lipopolysaccharide is a structure that is present in the gram negatives but not in the gram positive, which is this layer here. So the outer membrane in the gram negative is actually a lipopolysaccharide or LPS, while in gram positive it's the cell wall. Let's move on now to the second slide to discuss more details. I put this slide for you so that you can remember this very easily in a mnemonic format. So this is a gram positive. It's a positive cell, and it has a peptidoglycan layer which is thick, and it has a teichoic acid, so we can remember it by PPT. While gram negative is the lipopolysaccharide layer. It has an outer membrane which is an LPS. It's a negative, and you can pee it long. So this is just for you to remember. This is the peptidoglycan layer that I wanted you to understand. So if you can see here, this structure here, it's actually different. So this part here describes what you see in a bacteria, a gram-positive bacteria, and this what you see in a gram-negative bacteria. So this cell wall or a murine layer is actually very, very complex structure that is composed of N-acetylmurine or N-acetylmuramic acid and N-acetylglucamic acid, and all of them are alternating. It's attached to these peptides or polypeptides, and this is the main structure. And I don't want you to go through the details, but I want you to understand why this layer is very important for us as microbiologists. This layer is very important for us because we can actually synthesize or make antibiotics to kill this bacteria by attacking the synthesis or the making of this layer. And if we attack it, then the bacteria is dead. And that's a target for a different type of antibiotics called penicillins, cephalosporins, and vancomycin. You will learn these antibiotics later in this course. There is another important significant implication for this layer as well. Now we move on to the second layer that is present in gram negatives but not in gram positives, and we call it lipopolysaccharide, or for short LPS. So this is the outermost layer of the cell wall in gram negatives only. Gram positives do not have this layer, and we also call it endotoxin because an endotoxin is actually a structure that is toxic to us, but actually causing antigenic effect, which means that it will stimulate our immune system to work against it. And you will learn this into more details when you come to the immunology part of this course. But these endotoxins are very, very harmful. When they are released by the bacteria, they cause fever. So if your patient has fever and he has bacteria, you will immediately know, gram-negative bacteria, you will know that this fever is actually caused by the endotoxin. And which part is the endotoxin of this bacteria here? It's actually the somatic antigen, which is actually, we usually use this for identification of the bacteria. So if the bacteria has this structure, which is formed of three parts, the O specific polysaccharide is actually what we use to identify the bacteria, because we have specific antibodies in the serology lab where you add it to the bacteria, and once you see a reaction, you will know that this is a gram-negative bacteria. So we have specific antibodies for all these bacteria: E. coli, Salmonella, Shigella, and Vibrio. Don't worry about the names at this point, because all of these names will be repeated again and again, and you will learn more details about these bacteria. So another structure that is present in gram-positive, not in gram-negative bacteria, is the teichoic acid. So teichoic acid is another important structure, and it's important because it's again responsible for evoking a response by the immune system. So what you see here is like a fiber layer or a hair-like structure extending from the cell membrane, and we call it lipoteichoic acid, and some of them extending from the cell wall, and we call it teichoic acid, and they are only present in gram-positive bacteria. Why this is important? Because, as I said before, it becomes important because it produces a response. So when this bacteria, when it attacks the body of a human, it causes a septic shock. So septic shock is what we call when the blood pressure of the patient drops very dramatically, and it causes the low blood pressure. Sometimes there is also fever. The patient will need to be staying in the hospital to be treated for this shock. And this is all induced by this structure here, the small tiny structure. Not only that, but the same structures actually mediate the attachment to the mucosal membranes. So this means that this bacteria, once it invades, it will attach to the membrane. So what is the function of all these structures on the cell wall? It's a very rigid structure that keeps the shape of the bacteria and maintain it. It protects the bacteria from effect of osmotic pressure. So what we mean by osmotic pressure is that the changes in the salinity of and also the pressure outside and the inside of the pressure forces the movement of electrolytes. And in order not for the cell not to collapse, we need to maintain that pressure. And that is done by the rigidity of the cell wall. It will also take part in cell division, as we will learn in a minute. Also, it's a virulence factor, like we have learned before. So virulence means the ability of the bacteria to cause disease. So it's actually what is causing the septic shock. Part of them also is causing the fever and the hypotension or low blood pressure. So like endotoxins in gram-negative bacteria. Another structure, we call it protein M in certain gram-positive bacteria like Streptococcus pyogenes, protein A in Staphylococcus aureus. All of these are types of pathogenic or virulence factors that belong to the cell wall. And on top of that, they actually help to support other appendages that we will learn about in a minute. We call these appendages pili, fimbriae, and flagella. Also, because we said antigenic structures like LPS and teichoic acid will stimulate the immune system to produce antibodies, they are very important for evoking or provoking the immunity in our bodies. Okay, so we have gone through the cell wall. Now we can move on to the rest of the essential parts and non-essential parts. So what is the cytoplasmic membrane? So if you remember, we have discussed this part here. Now we come to the next part, which is the cell membrane, which is this layer here, the blue layer. It's actually composed of a phospholipid bilayer. Bilayer means two layers, so you can see them here actually in two or in pairs. What is the function of the cytoplasmic membrane? One of the first important functions is that allows the active transport of the molecules into the cell. It also allows the generation of energy by a process called oxidative phosphorylation. It, as we learned also previously, that this allows the synthesis or the making of cell wall. So the cell blocks, the cell wall, the blocks of the cell wall are made here, and they are exported for the cell wall to be made. Then the secretion of enzymes and toxins. So everything that is secreted or made inside, they are secreted through the cell membrane. Now let us move to the other structures. So I want to go through the remaining structures very briefly here. We have nucleoid, which I mentioned before. So it's the single circular molecule that the bacteria has. And on top of that, there is an extra chromosomal. So if this is the chromosome, there is an outer structure that is called the plasmid. And I'm sure that you have heard about plasmids before. So these are double-stranded DNA, circular structures, and they do not need the original DNA to replicate. That's why we call them autonomous, or they replicate independently. And they are important to us because they carry a lot of genes that causes the causes the bacteria to become resistant to antibiotics. And the bad thing is that also bacteria can like exchange these plasmids, like they borrow and take these kind of resistant genes among themselves. This is the picture of a plasmid when it's collapsed. And this on the top part you can see when it's relaxed or not super coiled. And you can see also here another structure called ribosome here, the small dots inside the cell. I mentioned this before, it's a protein synthesis structure. And in the bacteria we have these 70S ribosomal subunits. And again, these are good targets for some antibiotics. You will learn these antibiotics in the pharmacology part of this course. We have another important structure we call mesosomes. So mesosomes, these are the red part here. It's a structure that is essential for respiration or the breathing of the cell, and again the cell wall formation. So it's similar to the cytoplasmic membrane. And granules, see the picture here. The granules are the small dark dots inside the bacteria, and they are there because they store nutrients and also energy. An example of this is metachromatic granules that stain red in this picture here. When you put a methylene blue dye in one of the bacteria called Corynebacterium diphtherium. Now let us move to the next slide. Okay, now we come to the appendages. What we mean by the appendages is these extra structures. Can you see these hair-like structures? So one of them is pili, these little hairs outside, and these are proteins that are arranged in helical structure. We'll see in a minute what helical structure means. They are very short and straight if you compare it to these flagella, which are much longer. And the function of these pili is to attach to the human cells, and they also allow the conjugation. What we mean by conjugation is the exchange of the DNA between the bacteria. They actually be transported through these pili. Then a very, very important structure is the capsule. So the capsule is this beige layer outside. It's a polysaccharide layer, and the function of the capsule is that it is antigenic sugar composition actually gives the identification by serology typing. So which means that, as I explained earlier, when you have something antigenic, means that if you add antibodies, it will bind to these antigens and it will help you identify that particular bacteria. One of these reactions, we call it Quilling reaction, that you will learn later. It's also, this capsule is a virulence factor, which means that prevents the bacteria from being eaten by the immune system. There are particular cells called macrophages, and what they do is that they eat out all these bacteria by a process called phagocytosis. And the bacteria can remain hidden from these cells by this capsule. Again, this capsule is very important as synthesis of vaccines, because any antigen on this capsule can be taken and then put inside a vaccine to be able for the immune system to be able to produce immunity. And this is what is done through a bacterial vaccine called pneumococcal vaccine, which is against Streptococcus pneumoniae bacteria. Now, this structure also, the capsule, helps some bacteria to attach or adhere to the human tissue. And these attachments allows the process of infection to happen. So in this picture, you can see what I mean by the capsule. So it's, this cell is pink, the bacterial cell, and it's surrounded by a layer which is lighter in color, so it's a de-staining of the capsule. Now we come to the third structure in this slide, which is flagella. So what we mean by flagella is these whip-like structures that extend from the cytoplasmic membrane, and it has a various functions. Some of the bacteria will have many of them, some of them will have one, some two, and so on. And the function of these flagella is actually it helps the bacteria to move looking for food. So it will go towards food or chemicals, and this is a process known as chemotaxis. Again, this structure is similar to the capsule in that it helps us to identify bacteria because it has an antigen called H antigen, and this H antigen can help us in serology, which means that this is an antigen that will produce antibody as a response from the body, and then the reaction will happen, and this is used to identify a bacteria called Salmonella. So in this slide, I wanted you just to show you the different bacteria with capsule. Can you see the capsule here? It's actually the lighter color, and this is an fluorescent antibody stain where you can see these bacteria graying in color surrounded by a halo. That's the capsule. And again here, this is a gram stain where you can see the bacteria with the capsule. In this picture again here, we see bacteria inside with a lot of a lot of them here are surrounded by capsule, while some of them are not. So the difference between the two is that the ones with the capsule can escape these small cells here, PMNs or polymorphonuclear cells, which are part of the immune cells, and the ones without can be easily identified and eaten away by the cells. The ones with the capsule can escape and then they go into the bloodstream, causing an infection. So in this slide, it's a little bit complex, but I want you to look at those pictures here to understand that these are electron microscopy pictures that shows us the complexity of flagella in here. And you can see that they are helical in shape. On the down side of on the down part of the picture below here, you can see these actually causing the flagella causes the bacteria to look a bit different on the petri dish. They look a little bit like swarming or moving around the plate, and this is appreciated for a group of bacteria called Proteus. We will learn about these in more details about these in the lab. Now we come to some of these structures called endospores. Endospores are very important structures because not all of the bacteria have them, but they are highly resistant to heat, to killing and boiling, and that's why if the bacteria has these spores, it can remain alive for many, many years, even if there are no nutrients. So if you add a disinfectant like alcohol, for example, these bacteria will remain viable. Many gram-positive cells have them, like Bacillus species, Clostridium species, and this endospore, what makes it very strong and resistant to killing is actually it has a very hard coat made by keratin. It's similar to the keratin that we have in our hair. It's very thick. It helps protect the structure, and it remains, for example, if the bacteria lives in the soil and there's not enough nutrients, it can remain in the soil for so many years. And these, once the nutrients become available, they germinate. The term called germination means that they come alive again and they start reproducing. And in this picture, just to show you the different shapes, but I don't go through the details. Now let us move to the next slide. So this is the second part of this lecture, which is about the bacterial growth. How does the bacteria grow is actually or divide or multiply by a process called binary fission. So from the term itself, you can understand that the bacteria, single bacterium divides into two. So let's move on to the next slides to study this in more details. So this is a growth cycle here. What you can see from this picture here, we have a cell that is with a cellular membrane starts actually swelling and then it goes inwards. So the movement of the inwards of the cell wall and then the cytoplasmic membrane, it will squeeze the two parts of the cell and then the organelles start dividing and distributing between the two sides. The genetic material like the DNA again will start dividing into two and then each one of them will distribute among the two versions. Then these two septations will approximate each other and then result into the complete division of the bacteria. So you have to know that bacteria does not, like one bacteria does not give you one. Actually, it's exponential or logarithmic growth, which means that one cell divides into two. So the formula for that is n, which is one bacteria, for example, it gives you 2 to the power g, which is the generation time. Generation time is the time required for the bacteria to completely divide and result into two daughter cells. The generation time is also known as the doubling time, and actually I will give you here an example for you to understand it. So let's say you have an E. coli cell. An E. coli cell will, one cell of the bacteria E. coli will take about 20 minutes in order to give you two cells. And 18 hours is the generation time for another bacteria called Mycobacterium tuberculosis. So Mycobacterium takes much longer time, so it's almost more than half a day to give you one cell. So what is the practical implication of this? It's actually E. coli, very fast growing. I can get the results. If I grow it today, I get it tomorrow. Millions of bacteria. But Mycobacterium tuberculosis, because it's very slow, it will take me weeks, up to 8 weeks, to be able to see the growth of the bacteria. So generation time actually depends on so many different factors. They are almost always depending on the environmental conditions that you grow your bacteria in. So it depends on the pH, nutrients, and also the temperature, the osmotic pressure, and so on. In this table here, you can see if we apply this formula above, if one cell divides, it will give you two cells. Two cells will give you, again, if you divide them actually into on the times the power of the one generation, it will give you two. Two generations will give you 4. Third generation will give you 8. Fourth generation will give you 16, and so on. So this is what we mean by exponential growth. It's not straight line growth, it's exponential, which means that it will give you a curve that I will show you in the next slide. So this is what you see in the slide. Here is the exponential growth of the bacteria with different stages. So when you add the bacteria to the culture media or the place where it's supposed to grow, the initial stage or the first stage is called lag phase, which means that the bacteria at this stage is not dividing because it's utilizing the nutrients to initiate or start the metabolism. Once it kicks off, it starts dividing very rapidly in the second stage, which is called the log phase. So you have rapid cell divisions, and then it reaches the stationary phase in which the cell division stops. So the majority of the cells at this stage are not going to divide because as the growth progresses, cells stop dividing, nutrients will be depleted, toxic materials will start to accumulate, and this will not allow the bacteria to actually survive. So in the fourth stage is the death phase where almost all of the bacteria will not be alive. So the number of live cells will go down. Now let us move to the next slide. So what are the optimal growth conditions? What we mean by optimal growth conditions is that what are the best growth conditions? So if you want a bacteria to grow, you give it food or nutrients. You provide a good pH ranging from 6.8 to 7.2. You provide the optimum temperature, which is for the majority of the bacteria is 37, but this is not a rule. There are exceptions in some bacteria. And you provide the right atmosphere in which there's enough oxygen and enough carbon dioxide. This is actually what, how do we determine that? Depending on the oxygen requirement, we divide them into aerobic and anaerobic. Osmotic pressure is the pressure that allows the bacterial integrity or the cell not to swell or shrink. And actually, this is what, even when you do that at home, let's say you want to make these pickles, and you know that for you to make an mango pickle, for example, or your sisters or your mother making it at home, you will see that they will almost always, add a lot of salt because salt will kill most of the bacteria. So it's a way of killing a bacteria. It will add also a lot of oil on top and some garlic maybe, because oil will prevent oxygen from going to the pickle, so it will not spoil your pickle. So you should thank your mother for knowing this basic microbiology knowledge here. And the other thing is that you add a lot of acidic environment. So like for example, you add vinegar. Vinegar is very acidic. The pH of vinegar is below 4, so it's a very, very good preservative for most of the pickles, sauerkrauts, cheeses, and that's how you make your food or from spoiling or protect it from spoiling. Similar thing that what we do here in microbiology. Okay, so let's move on to the next slide. So in general, nutrients for the bacteria is media. And media is the combination of ingredients that will support the growth of the microorganism. Common ingredients of culture media are carbon and nitrogen source. So what you provide is these main components. You need energy source, so you have to make sure that you have good source for hydrogen donors and hydrogen acceptors like oxygen. You have to have minerals, so you can see here similar to human beings. We all need oxygen, all need nutrients, and also minerals like sulfur, phosphorus, calcium, magnesium, and trace elements. All of these trace elements are needed for the enzymes to act like catalysts or activators of enzymes. The fourth one is agar, and agar, what it means is like gelatin. So gelatin is a complex polysaccharide that we add to the media to make it solid, and it's made of agar or agarose, and agarose is a powder, and when you add water in it, so it becomes like gel. When it becomes solid, it solidifies and it gives you like gelatin or gel. So that's the end of this lecture. I think I've done very great with time. It's 49 minutes. Thank you very much for listening. And I'm sure that the majority of concepts you have also learned in the previous lecture, which was a bit of dense lectures. That's why I've decided to make this lecture a little bit less dense, because a lot of things have been covered in that lecture. So I really advise you to study these lectures together. Thank you very much. If you have any questions, please let me know by Moodle forums or even my email. Thank you.